All right, so today what we're going to do is we're going to start talking about how DNA kind of starts coding for a message. So DNA is genetic information that is housed in the nucleus of every cell in your body, and it should be the exact same copy in every single cell in your body. Should be identical. Because during cell cycle, mitosis, meiosis, G1S, G2M, during S phase, the DNA is replicated. And we're going to talk a little bit about that replication piece. A couple things to kind of reiterate from last week is Chargaff's rule. Every A there's a T, every T there's an A, every C there's a G, every G there's a C. And then there's three pieces of a nucleotide, which consists of a sugar, which if we're talking about DNA, the sugar is called deoxyribose. If we're talking about RNA, which is coming in tomorrow's guided notes, the sugar is called ribose. It has a sugar, it has a phosphate and a base, a nitrogenous base, an A, a T, a C, and a G. These are the bases that we're referencing all the time. And a nucleotide is what they call a monomer. It's the single unit of a polymer, which our polymer in this case is DNA. And the analogy that I use is the monomer of our classroom is one of these bricks. And one brick by itself doesn't make up a classroom, but if you put a bunch of them together, a lot of single units, a lot of monomers, we get a polymer, and our polymer would be our room if we're using that analogy. The nucleotide is a unit that's called a monomer, and if you put a bunch of nucleotides together, we get a nucleic acid. We either get DNA or we get RNA, depending upon a couple different things. Okay, so we're going to spend a little bit of time kind of talking about that. Kind of a big thing that we're going to be talking about today is DNA codes for a message. DNA codes for a message. And when we talk about DNA, this transfer of DNA, which I realize this is kind of hard to see, but the idea is we have this message written in DNA that codes for a particular protein. Ultimately, the idea is we are, I wonder if this is going to look awful from a distance. It might. Ultimately, what we're doing is DNA codes for a protein. It has genetic information that says this is the protein that's going to be made. And you have the hair color you have because of proteins. You have the eye color that you have because of protein. So you inherited the genetic information from your parents as DNA, but the reason why you look the way is not because of your DNA, it's because of the proteins that your DNA codes for. So the kind of the transfer of information is DNA goes to RNA, then RNA codes for a particular amino acid, and then you put a bunch of amino acids in the right order, and we get a thing called a protein. So the way that we kind of assemble these amino acids, the order in which the amino acids go, changes the proteins that we get. Okay, so when we talk about a nucleotide is the monomer of DNA, an amino acid is the monomer of protein. So you put a bunch of amino acids together, you get yourself a protein. But if you change the sequence of amino acids, there's 20 different amino acids. You change the protein, you change the message, you change your appearance. Okay. So that's kind of the where we're heading today. Some things that we've mentioned is we want to talk about hitting some of the highlights of your section notes. So your section 2 guided notes is something that we asked you to have completed for class today. And we want to go through and make sure you have a general understanding of this. So the idea of section 2 is on DNA replication. So why does DNA need to replicate? Because every single cell in your body needs the same copy of the DNA. Now, not every cell in your body codes for the exact same protein, right? Some of your cells have a protein that say be a blood cell and do blood cell stuff, okay? But another cell in your body still has that do blood cell stuff message, but it doesn't code for it. And instead, a different cell in your body says, do skin cell stuff, whatever that is. Okay, so every single cell in your body has the same code, has the same DNA. It's just some of those different cells produce different proteins based upon some other mechanisms. But they all have a complete set of DNA. Okay, so cell cycle, G1S, G2, M phase. We're talking about S phase right here. So during S phase, the DNA is going to replicate and make an exact copy. And remember that you are a eukaryote, and you have a nucleus, and all of your DNA is housed in the nucleus. And it's a very precious thing. It's a very important thing. It codes for a particular message, and it wants to be protected. The nucleus kind of protects that. The only time it's not in the nucleus is during mitosis or meiosis, where the nucleus dissolves. Otherwise, it's housed inside the nucleus. So this replication takes place in the nucleus. Okay, so you are a eukaryote, and all of this is fit really nice and packed tight into your nucleus. Now, if you were to take a cell in your body, one cell in your body, a single cell, you name the cell, skin cell, cheek cell, muscle cell, blood cell, and you were to take every single piece of DNA, you were to uncoil it and stretch it out, and then you take the next piece and you stack it end to end to end, one cell would have about 2 meters. I'm about 2 meters tall. Mr. DeBoer probably is 2 meters tall. Okay, would be my guess. I'm serious on Mr. DeBoer. Not quite, but closer than I am. Right, so about Mr. DeBoer. Okay, one cell in your body, a cell in your body, has about the same end-to-end amount of DNA for Mr. DeBoer. That's in one cell. Now, how many cells do you have in your body? A lot or a few?
A lot.
A lot. And I don't remember the number. It's in the trillions, though. And I don't remember if it's 30 trillion or 300 trillion. I don't remember the number. It's a lot of trillions. And then your video, what did it say? If you were to take every single cell in your body and take the 2 meters of DNA from each cell, and you put each cell's 2 meters end to end, it'd go from here to the moon and back, 37 trillion cells in our body, about. It'd go from here to the moon and back, what was it, like 600 sometimes? Wasn't it something like that? Wasn't that in your video for the weekend?
It was.
Okay. I think it was like 600 times your DNA in your body, if you took it end to end to end to end, would go to the moon and back many, 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 many times, hundreds of times. Okay, so why can it fit in why can 2 meters worth of DNA fit inside one cell, which is really small? Because it's really, really compact, and it's just wrapped around and wrapped around and wrapped around these things called proteins. So we have our double strand wrapped around proteins, which are then coiled again, and then those coils are coiled again, and they're coiled again in a fully condensed version. We see something called a chromosome. Okay. Remember Chargas rule, for every A, there's a T, every C, there's a G, for every A, there's a T, and every C, there's a G. Complementary base pairing, anti-parallel, it's upside down on purpose. These are hydrogen bonds in between. These are covalent bonds. Covalent bonds are strong bonds. Hydrogen bonds are relatively weak bonds. So why is that beneficial? Because we need to be able to break this for the replication. So replication is all about enzymes. There's 3 enzymes. I'm really more concerned about 2 of the 3. DNA helicase is the first enzyme. An enzyme is a specialized type of protein. We're going to be talking about proteins. We're going to talk about enzymes right now. Enzyme is something that's going to speed up a chemical reaction, does a particular chemical reaction. And DNA helicase is the thing that unzips or unwinds, is kind of how I think of this. I think of this as a zipper. I'm sorry, I think of DNA as a zipper, and I think of the helicase as a zipper pull, right? The actual thing you grab on your coat. Okay, I think of that as this. So this unzips the DNA. The DNA helicase is going to be the thing that unzips it. And now what we'll have is we'll have 2 separate pairs, complementary base pairing, Chargas rule. But the idea is helicase right here is unzipping our double helix, 2 strands, is unzipping it. And then a different enzyme called DNA polymerase comes in, and it's another enzyme. Now, polymerase makes polymers, and our DNA is the polymer. So what it does is it brings in the monomers called nucleotides, and using Chargaff's complementary base pairing rule, for every A there's a T and every C there's a G. And blues and greens go together, and yellows and orange, yellow and reds go together. And the idea is this enzyme brings in the complementary base pair to make the complement of it. Okay, now it goes in two different directions, and that's fine. But the idea is, if you kind of look, this sugar phosphate backbone is white, and that's trying to show you that's the original strand is in white. But then these free nucleotides are bringing in sugar phosphate that's kind of flagged or tagged as blue. So when we're done, we have a white strand and a blue strand that are all part of the new sequence. And there's a different picture that we'll show you in a second. The third enzyme, I don't feel as, I mean, it's important, it's maybe not as important in the grand big picture. It's another enzyme that kind of connects your sugar phosphate backbones back together wherever there's kind of a break because of the direction in which they go. But, so, helicase is the zipper pull, unzips the DNA. DNA polymerase brings in the complementary base pairs to form our polymer. DNA polymerase makes a polymer, and then ligase is going to go through and kind of stitch it back together. If we start with a DNA molecule that's in, I don't know, purple, magenta, whatever color this is, if that is the color of our original strand, is referred to as a semi-conservative model. And the reason is, if this is the original, after replication, it's somewhat conserved, or half of it is conserved. You could even say semicircle, right, half. So if this is our original strands that are represented by purple, because of how this kind of unzips and serves as a template, what we end up with is our new strands of replicated exact copies that happened in S phase. This is my original strand. Notice how it has one purple strand on each of my double helix, but then the blue strand represents the new nucleotides that were brought in. So it's not a direct photocopy of this, but it uses half of it to make one, and it uses the other strand to make the other, to make an exact copy, and I don't care about this part. But it's a semi-conservative model because half of it is going to be used for the next, to make the next phase, or to make the next copy. Okay? DNA replication, it's all about enzymes and Chargaff's complementary base pairing rule. A's goes with T's and C's go with G's. The original model unzips, and the new strands, the copies, are one side or one of the strands is the original, that it serves as the template for the other strand. That's DNA replication. That one actually right? That red was dying on you, I think, wasn't it? I think there might be another red one back there if you get on that table view. Okay, any general questions you have about DNA replication? Okay, then what we're going to move on to is we're going to move on to this. We are going to move on to this idea that DNA codes for a message. So when we have a coded message, let me rephrase it. Well, no, I said it codes for a message. That's fine. So if there's a message that's important, you want to make, and it's like, well, whatever. Let me start over. During the wars, there's a lot of times that messages are sent between allies with each other, but they don't want the enemies to know what's being said, right? Because if you say, hey, we're gonna move to this location at this time on this day, then the enemy could say, well, if they're gonna do that, then we're gonna do this, right? So if you kind of know the game plan, it's almost like cheating, right? So a lot of times they'll code their message, and there's some historical things that that we've heard about in World War II, that there's different types of coding that they use for message. Like the Navajo language was used as a coded message, that they had people that would, from the Navajo tribe, that would send messages in Navajo. But if you didn't speak Navajo, it didn't mean anything, right? So coded messages. But a coded message is only good if you can decode the message. Called like a cipher, I think it'd be, you have to be able to decode it. So the idea is we're gonna kind of model this, that DNA codes for a message. Okay, so the way we're gonna kind of go through and do this, oh shoot, you know what? Mr. DeBoer, I ran new copies of this because of the shift. I just didn't actually hit print. Should I go hit print on them and we can pass them back out, or is it not that big a deal? It'd be helpful, wouldn't it?
Yeah.
Okay.
Wouldn't be a bad idea.
Okay, can you kind of talk about start codons while I go and grab the 15 copies that I need?
Yep. So at the beginning, to get going with any of this, we're going to always begin with a start code, okay? Can you see this bottom part okay? With it being a little like that? So where do you see start? Right here. Now you'll notice we've got a first, a second, and a third. So up in the top, the direction said, code the following word using the MyDNA table. So this is the table, and this is made up. We need to begin the sequence with start. So what we're gonna do is we're gonna look first. So across here, this T, thymine, here's our start. First we go here, okay, so we're gonna write a T. Second, here's our start. We got the T first. Second, we have, or N. What letter? A. So we're gonna write an A in. So the first and second are pretty easy. The third is a smidge more tricky. And that's why Mr. Davies ran those other copies. So now I'm gonna look. In this box I have 4 options, right? Actually, I'm going to, yeah, I'm gonna switch.
All right, I'm gonna walk around, and that sheet that Mr. DeBoer has up on the screen, that bottom one, I'm gonna have you throw in this recycling can.
Yeah.
Okay? No, the full sheet, sorry, the full sheet. I'm gonna take the full sheet, and we're gonna recycle that. The alignment is slightly off, so because of the alignment being slightly off.
Yeah, you'll see why in a second.
Okay, so then, did I get everyone's? Now I'll grab a new one from right there. Okay, and you're gonna say, oh, it looks very similar. There's a slight shift. It's not perfect, Mr. DeBoer, still I think, but it's
Yeah,
better.
it's pretty close though.
Yeah. So,
So we got TA. So again, here's our start. First we looked here, hey, we got a T. Second, we've got an A. Third, I'm gonna note, you gotta pay attention to what row you're in. Now I'm going to come over to this column. Third, I go straight across. What's my third? C.
Yep. So TAC is referred to as, for us, this is coding off of DNA. Now there's lots of different tables out there that we can code off DNA, mRNA, tRNA, all these different things. This one's off DNA. But it's a start codon. Our start codon for this activity is a TAC. Okay, we need a TAC in order to start coding. And I don't know what Mr. DeBoer told you about start codons, but I'll just say this really quick and stop me if he said this. But your DNA, right, is 2 meters long. Every single cell in your body is like 6 meters or 6 feet tall. It's as tall as Mr. DeBoer. So we don't code everything. We are coding for very specific parts, segments of DNA And we don't know where to start coding until we hit the start codon. So we have this really long sequence, and we're scanning, scanning, scanning. Oh, start coding here. That's what a start codon is. Codon is just a 3-letter combination. So we have our start codon. Now, good kind of strategy. Notice how Mr. DeBoer kind of drew vertical lines separating the S and the the S and the T, because each letter is coded for by 3 base pairs. Okay, so right below that S, there's 3 little dashes, 3 little blanks, and that's for 3 base pairs, A's, T's, C's. So we need an S. So you gotta look in our table and we gotta find S's.
Let's see, we got S's right here.
Okay, now how many different S's are in our table? 3. Which one is the right one to use? The answer is they're all right. Okay, they're all perfectly fine. So what this table is saying is DNA codes for letters in our alphabet. This is just a model, this is an activity, an analogy that we're going to do. That DNA codes for letters in our alphabet so we can spell words. In our cells, however, DNA codes for amino acids. Okay, and amino acids code for proteins. So in our analogy, DNA codes for letters, letters combine to make words, right? DNA codes for amino acids, and the sequence of amino acids, right? The sequence of letters determine the word we're writing. The sequence of amino acids determine what protein we're making. So that's kind of what in the back of your brain we're thinking about. And we have 26 letters in the English alphabet. We have 20 different amino acids that we can use in the protein alphabet, I guess you could call it. Okay, so we have 3 different options for an S. It doesn't matter which one, pick one.
I'm gonna pick the bottom.
So Mr. DeBoer is choosing the bottom S. Okay, so of those 3, you could use the other ones if you wanted to, but let's all kind of look at that one. If we are coding for that bottom S, what is the first letter of our 3-letter sequence that we're gonna be using? What's the first letter? T, right? Because it's in that box, you chase that over to the left, that's a T. Okay? Then you look up for the second. What is the second letter going to be? A G. Now, assuming that we are looking at that bottom, the last of the 3 S's, what would the third letter need to be? A C. Okay? So the 3-letter combination that we chose is TGC. Now, what if, instead of coding for this S, what if we had picked this S? What would this 3-letter combination be? First letter would be a, the second letter would be a, and the third letter would be a T, right?
And this looks different from GGT.
But there's different ways to code for the same letter, just like there's different ways to code for different amino acids. There's some replicates that we can code for the same letter different ways. Okay? How many T's are in our little table? How many T's do we have in our table? 2. So how many different letter combinations can we get to come up with a T? 2. What's the first letter of our T, regardless of which one we pick? The first letter is going to be a
G.
T. The first letter is going to be a T because we trace it to the left first. Okay, so we got to go all the way left in that row. So the first letter is going to be a T.
Again.
What's the second letter have to be? A G. The second letter has to be a G because we go up that column and the second letter is a G. Now, depending upon which T you picked,
Top or bottom?
which one do we want?
Top.
Top. So the first letter is a T, the second letter is a G, and if we choose the top of those 2 T's, then our third letter is going to be a G.
But had you chose the bottom T, bingo.
Then it'd be TGT. Okay, so this just takes a little kind of practice getting used to, but once you kind of get a hang of it, it's not too bad. How many different A's are there? 3. The first letter has to be, because of the way they fall, the first letter of any of the three A's is going to be a, and the second letter of any A is going to be a A. Now, there's three different options, so top, middle, or bottom, which one do we want? Which one? Middle. So if we are coding for that middle one, now could you code for the top one? Sure. Could you code for the bottom one? Sure. They're all going to spell the letter A. They're all going to code for the letter A. But if we are going to go for that middle one, then the third letter would have to be a C.
Oh,
If it was the middle one.
it looks like a G from what you were
Oh
saying.
yeah, up on here the screen is kind of hard. But if it would have been the third one, then it could have been AAG. If we would have picked the top one, then it would have been AAA. There's different choices that all code for the same thing. Just keep that in mind. Why don't you try the letter R on your own? What's the three-letter combination that codes for the letter R? So if we're coding for the letter R, the first letter would have to be we have three choices, right? So the first letter would have to be G. The second letter would have to be now the three choices for the third letter could be GGA, if it would have been the top. It could have been GGC for the second, or it could have been GG. Right? Those are all different ways that we could code for the same thing.
I'm just writing the options up here. You would have picked a letter, so you would only have the three-letter combination.
Try the W if you haven't done so already. It's the three-letter combination that will get us to the W. Okay, so if it's a W, the first letter would have to be followed by a T, and then your two last letters could either be CT. What could be the third letter? Could have been a G, or it could have been a CT, T, right? Try the last couple on your own. Code for the A, then the R, then the S, and then find the stop codon and make sure you code for the stop. All right, so there are a couple different options that you could have had for that. You could have gone AAA for the A, you could have gone AAC or AAG For the R, it could have been GGA, GGC, or GGG. And then for the S, there's 3 different options. So depending of whether you chose these bottom 2 or this top, because it kind of spills into the different categories. You know, it could have been TGA or TGC. However, if you would have chosen this one, it could have been GGT. Would have also been perfectly fine. But the idea is we need a start codon to say start spelling our word now. And then we need a stop codon to say stop spelling our word now. So for our sake, what does our stop codon have to be? 3-letter sequence for stop is AAT, right? So we have to end our word with AAT to say stop spelling our word. So DNA, this is how we can code for a message. What would that be called? Encoding? We'd be encoding the message? Think, maybe? I don't know. But now we want to decode it. Now, this is going to write some word. We're going to use the same table. What does GAC code for? Before you write anything down, don't write anything down yet. What does GAC code for? What letter does GAC code for? What's that? GAC codes for letter G, A, C codes for the letter D. However, do you start spelling your word with the letter D? Do we start spelling a word with the first 3-letter combination that comes across? No, we need in order to know when to start, we need to have a start codon. And what's our start codon? TAC. We need to have a 3-letter combination of TAC. Are the first 3 letters in this row TAC?
Yes.
That says TAC?
No.
No.
GAC.
GAC, that is not a TAC. That does not say start spelling a word right now. So we only look at the first 3 letters. That is not a start codon. Okay, so we scribble out the letter G. Okay, and now we shift over 1. Is that a start codon? Is ACT the start codon? What's our start codon again?
TAC.
TAC. Is that say TAC? Nope. Scribble out that A. Shift over 1. Is that our start codon?
No.
Nope. Shift over 1. Is that our start codon?
No.
No. What are we looking for?
TAC.
TAC. So we can kind of just keep sliding over 1 at a time
Found it.
until we find, hey, look, there's my TAC. That's my start codon. I would give that TAC a circle. Okay? And then that says start spelling my word here. This is all, for lack of better words, term, it's called junk DNA. It's not junk, it's just non-coding DNA for this particular word. It might have some function, it might not, but it's not part of our word spelling. So we got to start spelling our word here, and then we look at 3 letters at a time. And notice how Mr. DeBoer kind of went through and chunked these off by 3s. Okay? And we are going to keep spelling our word until we get to a...
G.
Nope. When do we stop spelling a word? When we get to
A.
an AAT,
T.
right? Our stop codon. Now, in this particular case, it may not be the very last 3-letter combination. Maybe it's a little early. I guess we'll have to wait and see. The suspense. Okay? Now, what if the first letter is a C, the second letter is a C, and the third letter is a T? Which letter in the alphabet does that 3-letter code for? CCT. So C tells you it's in this row, right? C tells us all the way across here, okay? And then the second letter is this column, so where these 2 intersect. So we're now in this box. And then the third letter is a T, so our T is the last one So what is the letter? So what? J. Okay, the letter is J. Now, these are slightly shifted, right? These are slightly shifted. But CCT is, the first letter of our word is gonna be a J. What about AAC? A is gonna be this, first letter. Second letter, A, is this column, so we're now in this box, this choice. And our third letter, AAC, C is our second one down. So our second letter is gonna be A. Try. Let's do one more, I guess. Let's do one more again. CAA, we go C, and then we go A, so we're in those group right there, and our final letter of CAA is our A, so we come across, and that's gonna be right here, right? C, the first letter is C, the second letter, and then A is JAB. Keeps coding, decoding, until you hit a stop, and then you stop. If there's more stuff after that stop, it's not, we're done spelling the word. Is this a TAC by chance, or is that a C?
That is a C.
It says start again. It doesn't, it says stop and start.
Yeah, it's a C.
Huh. Well,
Oh, yeah.
whatever. Be a new word, sorry. Okay, what's the next couple letters? What's the next letter? So it's JAB, the fourth letter would be
C.
another B, right? And then what? An A, and then a stop. So what do? You stop spelling that word. Kind of pure coincidence in terms of random sequence letters typing. What's the next three say after that stop, actually? I know it said to stop, so you should stop, but just what does that tell us to do? Start. So what would we do? We'd start spelling the next word. It's just we're out of stuff to spell, right? But that's kind of an accident. Do we have them try this next one on their own completely? I think so, right?
Yeah.
Okay, decode this last word on your own. Remember, what are you looking for? What do you have to start with?
TAC.
Gotta find a TAC. So you look 3 letters at a time. Is that a TAC? If not, you shift over 1 to the right. Is that a TAC? If the answer is no, shift it over 1 more. I like scribbling it out like Mr. DeBoer did here, because then we say, don't look at that. Now we're only looking at the next 3 from here on out.
And I think we're 2 blanks short on this one, too.
Yeah, we are. You're right.
It fits if you don't write the start.
Oh, sure. Yeah, if you didn't write the word start and stop, you're right. It would fit the word really well.
Everybody get it? Or are we still working? I see people working yet.
So what's the next three letters spelled? You gotta start, what's CAT code?
Oh.
So TAC was, that start, right? So what's CAT code? Well, specifically though, which one, right? So C, A, and then T is specifically that one, right? So what's your next letter? Nope. C. That's CAT, what's ACT? Yep. So where's your start code?
I'm not sure if this is right or not.
So where is the start code?
Right here. I'm like kind of confused on how to do it.
What's the three-letter sequence that says start?
Right here, D, right at the C.
Look on that table. Where does it say start? Okay, and what's the first letter? Start?
F,
Nope,
or M.
nope, yeah, on this one. So that's the box we're trying to pull. So the first letter is the T. So T, what's the second letter? Second letter is
E, A.
A, so it's T. A gets us to this box, but
How
we
does
want
it
that.
go, so why is it going from here all the way up to here?
So this first letter tells us something in this row.
Mm-hmm.
Okay, well, we want specifically that. Okay,
Right.
so the first letter, or three, is the T.
Mm-hmm.
Okay, and now we want this column, so the second letter could have to be an A. Okay, so it's TA, and then we want that second spot, so we change that over as a C. So our start codon is a TAC. So where in that sequence is a TAC? Circle where you see the first TAC. Right there, yep, that's where we start. That's our start. So now what's the three-letter combination after TAC? What is CAT? So, CAT. Yep, so what's the CAT code for that? In here, so our sequence is CAT. Yep. Yep. So C tells us in this row, okay, it's somewhere along here, but the second letter is now an A. CAT code is that. So where's the second letter? A. Where's that specifically on the chart? Nope, yep, which tells us in this column, right? So the first letter says this row. What's that called?
Lily Lambert?
Yeah. So the first letter is this row, the second letter
So
is
what
this
you want,
column.
maybe you want maybe like check
And then
them off.
the third letter is
Put a-
specifically that T.
That was start,
comes
that was
across.
C,
So the second
That was
letter
H.
is C.
so now you're on GAT.
When I look right here.
Yep.
Okay. Oh, one more second. So what's the next one?
The next letter, starts
So yours. Nope, we're spelling the word, so your
Yes,
start.
there you go.
CAT said what?
And then C
It coded for what?
here.
Because we put that one together, so it'd be coded. CAT.
C, A,
letter of,
Why do I have to spell word?
because DNA codes for a message, and this is an analogy we're using to help you figure that out. So C says it's somewhere in this, right, and we're gonna- right there, C-A-T. My first letter is C.
Is that the one we're on?
Somewhere here. My second letter, second letter of C-A-T is an A. And somewhere in this column. So where does this row and this column intersect? This box of 4, right?
Oh,
And then the final letter is a T. My third letter is a T. We come all the way across. That codes for a C. That letter is a C.
this one
That
should
should
be,
be a C.
so C and C.
Well, you, which one, I think it was
Codes for the letter C.
you doing CAG,
This was- Look at the next.
right? So C,
So start from A,
Yep.
A,
and then go to C,
A goes here. And my second letter is a
C,
column of which one? Yep. And then my final letter of T.
so over here down to
A was here, and C is here. Four. Cross. All right, so Star Wars fans, if not, sorry, what does this spell?
how about the
Chewbacca. Okay, this is gonna spell Chewbacca. DNA codes for a message are DNA sequence codes for the letters for Chewbacca. So we go through and we spell Chewbacca, we keep going, we keep going, we keep going. You're one letter blank here, yeah. And then we have a stop, right? What's our stop? Our stop is AAT, so AAT is right here. There is another 3-letter combination after that, but I stop spelling my word because it says to stop. Okay, that's where it says to stop. All right, so what we need you to do is now we're gonna code for your name. Okay, so in order to spell your name, we first have to code to say, let's start spelling your name. Everyone needs to say, start coding your name. Because on Wednesday, we're gonna do this. You're gonna code your name in a DNA sequence. So everyone needs to have a TAC, right? Because that says start. T, A, C is my start codon. Then insert your name. Each one of these blanks up here on this chart represents a letter of your name. Okay? So you go through and you spell your name, and then how do we know when to stop spelling your name? You put a stop at the end. Okay? Then the combination, and we are running really tight on time here. The combination is going to be then what 3-letter combination would code for the first letter of your name? 2 things we want you to have done before you come to class on Wednesday. One is to have your name coded for. Okay, that's one thing. The other thing, section 3 guided notes. Section 3 guided notes are there. The videos are posted. I really would encourage you to watch the video on this one. There is a slide presentation as well. There's a lot in section 3. I'd encourage you to watch the video on section 3, but it's up to you. 2 things. Have your name coded in the DNA sequences, as well as section 3 guided notes before you come to class next time.
All right.
Have a great and wonderful day, everyone.
If you need help, come find us.